Happy to have Emily Josephs with us today. She's at Michigan State University, where she will any day now be an associate professor. You're all very happy for her. Em has been at MSU for 6 or 7 years now, and before that, she was at the University of California at Davis doing a postdoc with Graham Coop and Jeff Rotavara, and then before that, she was at Toronto working with John Stinchcomb and maybe a little bit of Stephen Wright, Stephen Wright, sorry. All of them very big names in population and quantitative genetics. It's not only the names that she's inherited. Emily has done a really amazing job at kind of moving the field forward in understanding how the environment and genetics contribute to our understandings of how populations adapt and change through time. She's also an amazing collaborator. She has 2 very long-standing collaborations, even at NC State with Seema Sheth and Rowan Rajan. She's the type of person who everybody wants to talk to. She's kind and insightful, and I'm very, very happy that she's with us today. So she's going to talk to us about her work on phenotypic plasticity, and then she'll hang out after the seminar for about half hour if you didn't have a slot with her. Thank you.
Well, thank you for that intro. I don't know how to like maybe someone else who actually fits the description. But thank you so much. It's been really cool to be here at North Carolina State. Like, I've never been to this campus before, but like I've collaborated with Ruben and with Seema, and I've known Rafa for a long time, and like it's just such a cool community of both like really great evolution and really great plant people here. So it's like really a treat to get to talk to all of you today. And I wish I could have stayed for like 5 days. Okay, so I will tell you this. So the really big picture that my lab is interested in is understanding why there's genetic variation for traits within species. Because when we look, across nature and all these different systems, we see lots of variation within species, where we were thinking about color, size, shape, or like history, timing, or anything else. And so we're really interested in understanding why does this variation exist and persist in nature. And to take it away from all the pretty pictures to cartoon worlds, as an evolutionary geneticist, what I mean when I say that is what are the evolutionary forces that shape genetic variation for traits? So I'll walk through some of the hypotheses we have for this, because we often set them up sort of in opposition to this idea that there's some optimum phenotype. So for these cartoon plants, you might imagine that there's an optimum size. In selection, if there's an optimum, we expect selection to move everything towards that intermediate size. And so the question we might ask is, why is this not happening? Why do we see variation and not just everything existing at this optimum? So our first hypothesis is just that we're wrong. There isn't actually an optimum size. Maybe we have plants of varying sizes because it doesn't actually matter what size you are. There's no correlation with fitness. This is always like our neutral or null expectation. Our second hypothesis is that there is an optimum size, so being too big or too small is bad, but that mutation is still strong enough to create variation. And so we still see variation that exists at a balance between mutation, which creates it, and selection, which removes it. And this means that the variation that we see is mostly deleterious. Often, the alleles that affect it will be present at low allele frequencies because they're having deleterious effects. And we call this mutation selection balance. Our third hypothesis is that balancing selection maintains variation. So maybe selection is actually acting to perhaps select for big plants one year and small plants another year. Or maybe, if everybody's big, there's some advantage to being small, or if everybody's small, there's some advantage to being big, which maybe doesn't make sense if you're thinking about plant sizes, but if you're thinking about, like Drosophila behavior with rovers and sitters, that's something where we do expect to maybe see some of negative frequency dependent selection. And so balancing selection is really a big catch-all hypothesis for any process where selection is increasing variation. Finally, there's sort of a special form of balancing selection called local adaptation, where the optimum trait differs in different places. And so we see variation present across a landscape because in some places it's more optimal to be big and in other places optimally small. So this can start to get more and more complicated as you imagine Now you have to think about migration between populations, contributing to variation and things like that. Okay, and so for all of these processes, you can think about variation resulting from this sort of big wheel where genotypes exist, they create traits which have variation, that creates the variation in fitness. The variation in fitness causes selection, which then changes the genotypes present for the next generation. And this is sort of the model that we sort of have underlying all of these different processes that can create variation. But one thing I've been really interested in since I started my lab is the role that the environment plays in all of these processes. So for example, the environment affects how genotypes determine traits, and we call that phenotypic plasticity. So the same genotype can create different traits depending on the environment. And then the environment also affects the relationships between traits and fitness. It may affect the relationship between fitness and genotypes, but we're not going to think about that today. And so I think that in order to understand these processes, we really need to think about the role that the environment plays in each step in order to understand why there's trait variation in nature. So I think I'm not the only one that cares about plasticity. Like most or maybe all biologists care about plasticity because we're all studying organisms in an environment, even if that environment is your lab or a growth chamber or something. And so these are just some cool examples from your department of people studying plasticity. Folks like Seema Sheth, who think a lot about how growth rate changes with temperature and how that relates to evolutionary processes. Work from Katie Smokowski-Heil's lab, thinking about how recombination rate changes with temperature. Work from Maggie Wagner, who is here working with a bunch of you, thinking about how heterosis changes depending on the microbial environment. These are all like examples of how, even if you're interested in different things, plasticity is really, really important to what everyone is studying. Two additional things I wanted to highlight is work from Rafa showing not just that there is plasticity, but that the genes involved in plasticity are in different parts of the genetic network, depending on the stress, which is always really cool. And then really cool work from Joe Gage looking at how selection affects G by E over the course of domestication, G by E just being genetic variation for plasticity. So there's been selection over domestication to reduce plasticity in maize. And so I haven't been able to capture everyone in the department, but hopefully I can convince you that many of you care about plasticity for lots of different reasons. However, I think it's been kind of tricky to study the evolutionary genetics of plasticity. And so I'll just walk you through like my simplified hypothesis for why. So population genetics really developed around ideas of simple fixed traits that were tractable to study, whether that was things like Drosophila phenotypes. These are actually Capsella leaves that were used by some early sort of evolutionary geneticists to study variation, or people looking at spatial variation in blood type frequency. It was, most of these ideas were around traits that were fairly static, that could be studied really straightforwardly. Meanwhile, it's been a really long tradition of evolutionary ecologists studying plasticity in systems that were not necessarily genetically tractable. So people know a lot about how different plants evolve in different environments, but these were things that wasn't really easy to study genetics. And so over the last few decades, both better genomics tools and some really smart scientists have started to really do great jobs connecting population genetics and evolutionary ecology. This is an example of a project that I worked on as an undergrad. So when I got my start in science, looking at variation in plasticity in Arabidopsis thaliana and linking it to genetic variation. But even this project took quite a long time to get from collecting the data to finding loci associated with plasticity. And finally, I'll just say that we're increasingly aware of the importance of plasticity because climate change is expected not only to change means, but also to increase the variability in climates. So this is just a map of summer temperature variability expected. And so that means we can already think that if the environment becomes more plastic or more variable over time, that thinking about how organisms respond to that environment is going to be a really important component to predicting what will happen during climate change or mitigating that response. Okay. Finally, I just want to put a pitch that plasticity is really confusing. And I think that's another reason why it's often really hard to talk about. Even when we say the term plasticity, sometimes I talk to people who think I'm talking about like genomes that can change an architecture really easily, or like something about the brain, like brain plasticity or neurotypical plasticity. And so just a few reasons it can be really confusing. So there's active and passive plasticity that people talk about. An example of active plasticity would be these thorns that acacia grow in response to herbivory. The idea being that after an acacia plant gets munched on, it grows thorns that then make it less palatable to an herbivore in the future. This is something where it's hard to imagine how that could happen or evolve through any reason other than some active selection for that response that is then creating that response. However, if we think about plasticity just as any sort of change in trait due to the environment, there's also lots of plasticity that can be passive or is just the result of some constraint of the organism. So these three plants differ in their response to drought. And this is not necessarily the plants making an active choice to do something different, but that they're better or less able to maintain, their growth or their fitness in the response to drought. And so some people think a lot about active versus passive. There are lots of reasons why you might care. But for today, I'm going to ignore the distinction and just think about plasticity as any change in response to environment, whether it's active or passive. Second, I think it can be confusing because we often, those examples, like the acacia example, we often think about plasticity as something that's beneficial. Our organisms are smart. They're seeing an environmental challenge and they're responding to it in a good way. But plasticity can also be maladaptive. And so, for example, what you can't see is that under this snow are like hundreds of Arabidopsis plants. This plant made the choice to flower at a specific time. Assigning agency to the plant it doesn't have, but this plant decided, flowered at a specific time due to some stimulus. It got the wrong information. It snowed a bunch on the plants, and it's not going to have great fitness because of it. It did make a few seeds, you can see. And so often plasticity is not beneficial, and I'll talk more about that later on in the talk. Finally, it can just be really confusing to measure plasticity. So we often use a reaction norm, where on the x-axis, we might have two environments, and the y-axis is our trait. And then we think about how different genotypes respond to different environments. This is work from a graduate student in my lab, Sophie Bicey, who's been looking at Arabidopsis from Sweden and Italy, using growth chambers to simulate current conditions in Sweden and future conditions in Sweden after climate change. And so by plotting out these reaction norms, we can then test to see if these slopes are different. In this case, they're not. So both Swedish and Italian plants have similar responses to the future environment in terms of they both flower a bit later in the future. But there isn't genetic differentiation between them. However, sometimes you see differences in how these plants respond. So now the y-axis is specific leaf area, or a measure of how thick the leaves are. And you can see that while both plants have reduced specific leaf area in the future, the Italian plants have a much stronger response than the Swedish plants. So we call this G by E. So there's genetic variation in the plastic response to the plant, or to the environment in these plants. However, this works really well with two traits or two environments, but once you start to get into more environments, it can be really, really complicated to measure plasticity. So if you have multiple environments, you might end up with strange curves that then it can be hard to interpret. Sometimes people like Seema's lab do really nice things sort of estimating those curves. There's lots of different strategies for doing this, but it can be really complicated and tricky to figure out the right one. So today I'm going to tell you 3 different stories about what my lab has been working on in terms of trying to get at how plasticity relates to the maintenance of variation within populations. And then if there's time, I'll tell you a bit about some of the other projects we're working on. I'm not sure there will be. Okay, so first, I'm going to talk about that question I mentioned earlier. Is plasticity beneficial? So as I mentioned before, environmental responses or plasticity can be adaptive. A classic example of this is the shade avoidance pathway, where when you take plants and grow them in the shade, they display this sort of classic response where they grow really quickly and a higher height than they would otherwise. The idea being that they're trying to really invest in growing taller than their neighbors so they can get more sun and energy and have higher fitness. However, if you're a plant that lives in the shade of a big tree, perhaps, showing the shade avoidance response is not actually going to be that beneficial because you'll never grow taller than the tree that's shading you out. And so, in fact, like many woodland, understory plants have lost that shade avoidance response. So we know a lot about this trait because of some really nice manipulations done by folks like Susan Dudley and Annie Schmidt to actually show their fitness effects of these responses. So they took plants and elicited the sun response and the shade response. And these are patients' plants, patients' plants. And then after they'd elicited that response and it was stable, they transplanted them to shady and sunny environments and showed that in the sun environment, the plant with the sun response had higher fitness than the plant with the shade response. And in the shady environment, the plant with the shade response had higher fitness than the sun response. This is really clear evidence that that shade avoidance pathway is adaptive in these plants. However, a lot of traits aren't really tractable to this type of manipulation. So for example, this is a plot showing gene expression plasticity in maize, looking at genes that change their expression in response to cold or in response to heat. We get thousands of genes that change their expression in response to these stresses. And these changes can be, ephemeral. So a plant might change in response to something, but it might not keep that response up long enough for you to then quickly move it to an environment, let it sit there and see if that response has changed fitness. And so I was really interested in trying to understand, how can we figure out if these types of expression changes are beneficial or deleterious? And what are some ways we could do that since we can't do what others have done? And so to do this work, I collaborated with Gina Balcom and Megan Van Etten at the University of Michigan. And they've been working on, and Gina's lab in general has been working on morning glories, which are both beautiful. I have some in my garden, but also an agricultural weed that can be quite a problem. I think actually here in North Carolina is one of the places where they've worked on them. And so they had done some really nice experiments with morning glories where they collected morning glories from a farm in Georgia that had not, or from a field site in Georgia that had not experienced any herbicide. And then they selected for herbicide resistance in some plants and control, and like they did controls where they didn't do any selection in other plants. And I should note that some morning glories have evolved resistance to the herbicide glyphosate and some haven't. These plants had not yet. After 3 generations, they were able to show that the glyphosate resistant plants did evolve some resistance to glyphosate. And then they did a treatment where they sprayed some of their plants with glyphosate. And then they looked at gene expression and the leaves of all of these plants. So they had data about what the expression of resistant plants was without glyphosate, the expression of control plants without glyphosate, and the expression of control plants that had been sprayed with glyphosate. So the first time they experienced this stress. And they were interested in trying to understand the pathways involved in the evolution of this resistance. But I realized looking at this data that we could zoom out and actually use this to understand, is this expression plasticity in these control plants adaptive or maladaptive? Because what we have here is information about plasticity that naive plants experience when they see a new stress. And we have information about evolution. How has expression changed during the evolution of resistance due to selection? With the idea being that if these changes tend to go in the same direction, we can infer that that response is probably beneficial. If they go in opposite directions, that suggests that the response is maladaptive. And so we took their data and plotted it out like so, where on the x-axis we have the plastic response, and on the y-axis we have the evolved response. And so over here means that gene expression increased in response to herbicide. Over here, it decreased in response to herbicide. Over here, it decreased in response to selection, and here it increased in response to selection. And the idea is that each point is a gene, and if you have something in this quadrant, it means that the plastic response was to express the gene less, but that the evolved response was to express the gene more. And so this is consistent with maladaptive plasticity. The plastic response goes in the opposite direction of the evolved response. But if you have a point here where the plastic response and the evolved response are both positive, expression increases in both cases, this would be consistent with plasticity being adaptive because the plastic response does the same thing that selection does over multiple generations. And so when we looked at all of the genes where we had evidence of significant changes in response to herbicide or response to selection, we see that genes show all kinds of patterns, but that overall, about twice as many genes were consistent with adaptive plasticity as with maladaptive plasticity. So that means that for 2/3 of the genes that we looked at, they had plastic responses that were in the same direction as the evolved responses when we selected for herbicide resistance. This was like actually really, really surprising to us because we were not the first people to do this kind of experiment. So previously, Cameron Gollenberg's lab had done a similar project using guppies, where they had taken guppies and exposed them to predators and then looked at gene expression changes in the brain. And they compared that plastic response to evolved responses in different predation environments. And they found that most of the expression changes in the brain were consistent with maladaptive plasticity. Similarly, work by one of my grad school cohort mates, Yuhan Huang in Drosophila, had found that when he compared plastic responses to cadmium and salt in Drosophila flies to evolved resistances in those sort of stressful environments, that most of the initial responses were maladaptive. And so we had been going into this expecting, I had been expecting to see mostly maladaptive plasticity because that's what other people had found, but we saw the opposite. And so we're really still curious to think about, why do we see that adaptive plasticity is so much more common in morning glory in response to herbicide than in these two other examples? And obviously 3 is not a lot of examples in order to make this kind of conclusion. But one of the things I can sort of hand wavy say that we've thought about is like, first of all, one option is just that plants are better than animals. Yeah. I don't think that's a great answer. Another one is probably, obviously these were all different kinds of experiments. They're different kinds of stresses, different lengths of selection time. Those are all probably important. The hypothesis I like the best is that the things you do to evolve resistance to glyphosate are similar to the things that plants do to evolve resistance to other types of stresses. Because a lot of these things, the genes we were looking at, were not actually involved in the glyphosate detoxification pathway, but we think may be involved in other types of processes that might prevent glyphosate from getting into the leaves, or quickly, things like that that might be important for all sorts of stress responses. And so Gina and I are following up on this by thinking about sort of correlations in stress response across different types of stresses. But we haven't delved too much into that yet, so stay tuned. Okay, so that was the first story I told you about. Is plasticity beneficial? And surprisingly to us, in this case, we found evidence that yes, expression changes are consistent with plasticity being beneficial or adaptive. So next, now that we feel really great about plasticity, why aren't all organisms able to evolve adaptive plastic responses to stresses? Just this. Okay, so this is a pretty big question. What limits the evolution of plasticity? And so I like to describe this thinking about a Swiss Army knife as sort of your ideal plastic organism. Any sort of problem that you encounter, you can sort of pull out the right tool to sort of solve the problem with the Swiss Army knife. And so why don't we have Swiss Army knives that can solve every single problem in our organisms? So the first hypothesis that people often make is that there are costs to plasticity. So perhaps plastic individuals have lower fitness than non-plastic individuals, even when they express the same trait, because maintaining that plasticity is costly. And so you can imagine this like having to have all this stuff in your handle makes you a worse knife than if you're just a knife. So you might opt, this knife might have higher fitness for cutting things. The other hypothesis people often make are constraints. There may not be a physiological way for optimum plasticity to evolve. So if you want to split wood, there's no way to fit a hatchet into your standard Swiss Army knife. And so this would really limit the evolution of plasticity to wood splitting. The one thing that makes this particularly confusing is that the data you would use to test these hypotheses is actually different. This is a hypothesis about fitness differences between individuals that are expressing the same trait. This is a hypothesis about something about like physiology or like response to selection. One thing that I was really interested in thinking about was this third hypothesis, which is about the accumulation of deleterious mutations. So imagine that you have a Swiss Army knife, but you haven't actually used the knife component for a long time. You wouldn't notice if it had broken or degraded because you hadn't like pulled it out to look at it. And so over time, you might end up having different components of the Swiss Army knife break if they're not often used, which seems like a little hard to imagine perhaps, but this is what's actually happening within natural organisms. Every generation, DNA has to be duplicated, and you can have mutations that occur. Selection can act against deleterious mutations that affect phenotype, but if a mutation doesn't actually affect the phenotype because it's involved in some plastic response that isn't getting elicited in the environment, then that mutation can spread and reduce the ability, over time, and potentially degrade the plastic response. And so the idea here is that you can imagine, at mutation selection balance, you always have new mutations that are occurring, filling up a pool of deleterious variants, and the negative selection or purifying selection removes that variation. But if negative selection is not able to act very often because these traits or these genes are only expressed in certain environments, then that pool of deleterious variants can grow and grow and grow and eventually degrade that response. And so this was not just my idea, but something that Mike Wade and Dave Van Dyken had proposed in 2010 as something that could limit the evolution of plasticity, but no one had yet tested it with any data. And so Miles Roberts joined my lab in 2020, which was a great time to start grad school, and for many of you started time to start too. And we realized that while we couldn't actually meet in person, we could test some of these ideas using publicly available data. And so Miles did just that, trying to see if negative selection is weaker on genes that are only expressed in certain environments in Arabidopsis thaliana. And so what Miles did was he took all the public RNA-seq data available for Arabidopsis, which was 27 terabases of data. He likes to put this in context of I think there are there's essentially 3 bases here for every fish that lives in the ocean. 27 terabases of data. So it was a lot. He then remapped that all to the Arabidopsis thaliana reference genome and quantified expression so that if we were looking at expression between different environments, we knew that wasn't due to different statistical or data analysis changes made by the scientists. And just to show what this looks like, he could calculate treatment specificity. This is just for expression in leaf tissue, taking all of the different data about the different environment that expression was collected in. And for each gene, he just measured treatment specificity. And what we can see was he actually found that a lot of the genes in Arabidopsis thaliana do show treatment or environment specificity. They're not constitutively expressed across every environment. Next, Miles wanted to look at selection on these genes. And so we took alignments of Arabidopsis thaliana and its close relative Arabidopsis thorata. And in every gene, we counted the divergent sites and counted up how many were non-synonymous or affected protein sequence change and how many were synonymous. With the idea being that, mutations have occurred since their common ancestor. Things that are synonymous are not selected for. Things that are non-synonymous are overwhelmingly going to be deleterious because it's easier to break something than to make it better. And so we could use the ratio of non-synonymous to synonymous differences to understand the strength of negative selection. So essentially the results are this big partial correlation table because lots of stuff affects negative selection. And so we looked for correlations between our non-synonymous divergence, synonymous divergence, which tells about mutation rate, treatment specificity or environment specificity, average expression, because that also matters a lot for selection, GC content and length. And then, so that's we're looking at all of these tables and looking for a relationship between non-synonymous divergence and treatment specificity. And that's exactly what Miles did find. There was a significant correlation between non-synonymous divergence and treatment specificity. This is something where 0.13 may or may not look impressive to you, depending on your scientific background. But we've known for a long time that there's a really strong relationship between non-synonymous divergence and average expression. And this is something that pretty much since people started measuring gene expression, and that the treatment specificity effect is about half the size of this really well-known long-standing correlation. You can also think about it, just thinking about the numbers, the top 25% most treatment-specific genes have more than twice as many non-synonymous diverged sites or non-synonymous substitutions as the bottom 25% treatment-specific genes, and the ones that are expressed all the time have many fewer deleterious mutations accumulating in them. And so overall, this pattern is consistent with the idea that deleterious variants accumulate in these genes that are environment specific, so involved in plastic responses. Consistent with the original hypothesis that the evolution of plasticity could be limited by the fact that these deleterious variants aren't being removed from the population as frequently. And so we have a lot of future questions from this. The first is we haven't yet made a link, how important are these deleterious mutations actually for organism fitness? And then based on that, then how much does this process prevent the evolution of plasticity compared to some of the other hypotheses I talked about? Additionally, like, your stats 101 professor would say, correlation doesn't imply causation. It's possible that it could go the other way. Maybe genes were already under weaker selection for lots of different reasons, and then they were then more free to evolve plastic responses. Finally, a former postdoc from my hallway, Sunil, he had the idea that maybe methylation affects both plasticity of expression and mutation rate. So he's investigating this in his brand new lab at Riverside. So I'm excited to see how that goes, because I would never have thought about that one myself. Okay, so to take it back to our main questions, we talked about why aren't all organisms plastic, and one potential hypothesis is that plastic responses just accumulate lots of deleterious mutations because they're not expressed all the time. Okay. So everything I've told you about so far is stuff that happened using publicly available data, and that's because I founded my lab in 2019. So I'm going to move on to telling you about some of the stuff we've been able to do since we got back into the lab, and which really centers around trying to understand plasticity in the real world and using data that we have generated ourselves. In the wonderful plant Capsella pastoris, or shepherd's purse, it looks a lot, it's often easiest to sort of introduce it in contrast to Arabidopsis thaliana, because they look so similar that sometimes in my department, where there's a lot of molecular biologists, they see people from my lab in the hallway and they're like, oh, what accession of Arabidopsis is that? But so Capsella, it has these really cute heart-shaped fruit. It's also far more common. So if you think you've seen Arabidopsis growing and you weren't like outside the greenhouse, it's probably Capsella and not Arabidopsis. Capsella pastoris is also an allotetraploid, so it has genomes of 2 different species that have been smooshed together, but we won't talk about that much. Capsella pastoris is a worldwide distribution. It's sometimes called the most common plant in the world. Its parent species are in Central Asia and Europe, so we think it originated somewhere around here, but it's since spread throughout Eurasia, and in the last couple hundred years to the Americas and Australia and New Zealand as well. So this is just an iNaturalist map, but it really is like everywhere there are people and it's not too hot. It's particularly common in Lansing, Michigan, where I'm from. Beautiful Lansing. And it loves Lansing because it loves really disturbed urban environments and sidewalk cracks. And we have a lot of sort of like urban decay in Lansing, Michigan. Particular population. There's just a picture of some of the populations that we have really locally available to us in Lansing. Yeah. Okay, so in 2020, I started collecting Capsella pastoris just when I was walking around my neighborhood because it was everywhere and I was really curious about it. And so these are all the populations I collected. There's no scale bar, but it's about a mile from end to end of this map. And the reason I was really curious about it is that there was a ton of flowering time variation in Capsella. So I was seeing that plants, for example, the tattoo shop population flowered over a month earlier than this muffler shop population. And these are just across the street from each other. And so I was really curious, is this due to microenvironmental variation? There's a lot more sun and heat at the tattoo shop. Or is this genetic differentiation happening at a really small scale? And so I collected all these seeds, and then when we got back into the lab, Mia Stevens, a tech in the lab, grew all the plants up in a common environment and looked at flowering time variation in these plants. And so this is just a picture of the plants growing in the growth chamber, and it kind of gives it away that even in the common environment, she saw lots of genetic differences in flowering time between the plants. And this is the data quantitatively. Each of these points is a different plant from each of these populations. And on the y-axis is days to flower. And what we see is that there's tons of very genetic differentiation. And I'm saying genetic because we're growing them in a common environment. There could still be maternal effects. But we see that there's lots of differentiation between different genotypes. And this y-axis goes from 30 days to 90 days. That's about the difference between like Arabidopsis from Southern Europe and Arabidopsis from Northern Europe. just happening within our like weird neighborhood of Lansing. So we're super curious about what's contributing to this variation and why it exists. And so to talk a bit about that, I'm going to take you to a like slightly more interesting city, New York City, because we just got our sequencing data back for the Lansing populations and haven't looked at it. We're not confident in presenting it yet. But we do have some older data of genomic sequence from 21 populations collected across the New York City area. This was collected by Rebecca Pankow, and she was a postdoc in my lab, and she was a graduate student in New York City. And she we also saw lots of phenotypic variation in Capsella and got really interested in it. And so she started collecting populations from across New York City, trying to get, gradients from urban to suburban areas. Then Adrian Platz and Maya Wilson-Brown worked to extract, so Rebecca did all the extractions and they analyzed all of the sequence data to try to understand, why is there genetic variation within New York City that contributes to trait variation. All right, so first of all, we wanted to know, how did all these plants get here? So one example of another New York City piece of wildlife are rats, who were introduced, they think, once to New York City based on this principal component plot. So these are genetic principal components now of rats. The colors are all of these different populations of rats around the world. All of the New York City rats cluster really closely together in this one area, which suggests that there was one introduction of rats to New York City. So we were curious, are the Capsella pastoris plants like rats? And the answer is sort of. So this is a PCA plot of all of our worldwide Capsella populations that we had available to us, and then our New York City metro populations. So you can see that they do cluster together with lots of Northern European populations of Capsella. And then when we zoom in, this is just showing the European, this is just a PCA plot of only this cluster of plants. And so when you zoom in, is you can see that all of these New York City metro plots do cluster together, suggesting one introduction, but we still do see quite a lot of genetic differentiation between them, which is pretty interesting. I will note that this little dot right here that's closer to Europe is from New Jersey. So we're still trying to understand. So right now what we do see is potentially consistent with one introduction. So suggested or introductions from only a few places. And we need to do some more careful demographic work to follow that up. So that suggests that the variation doesn't necessarily exist from like what we thought might have happened, which was that plants were coming from multiple places to these cities. At least for New York City, it might be different for some of our other cities. So we've since then gotten excited about this and been collecting plants from around the Midwest and the West Coast to try to better understand how these plants arrived to North America and then what they've been doing since they got here. So some of our locations are very scenic in California and some of them are in direct parking lots. It's kind of fun to do. So since we're still trying to understand sort of the genetic history of these plants, I wanted to change gears and also tell you a bit about some of the traits that we're interested in. And so one trait that people have been working on for a really long time in Capsella pastoris is leaf shape, because they show a ton of leaf shape diversity. And so Asia Hightower, a graduate student in my lab, has been working really closely with Dan Shitwood, who studies leaf morphology. So they've been working to model Capsella pastoris leaf morphology in order to understand how it evolves. And this has actually been quite complicated, it turns out, because they have variable numbers of lobes. So Dan was really excited about it because it meant they had to come up with some new techniques to describe the lobing. That's about all I know about that. You'd have to talk to him about more. So then Asia her pandemic project was taking these digitized herbarium samples of leaves and tracing them and then using them to try to look at leaf shape variation across the range of North American Capsella pastoris. So she traced hundreds of leaves from all across North America using these herbarium samples and worked with Dan to try to quantify shape variation and then figure out why what environmental factors might contribute to the shape variation. And so what Asia found was that temperature during development mattered a lot. So this is the temperature of location the leaf was from for the 3 months before that sample was collected. Each of these points is a different Capsella plant. And what we found was that there's plants from warmer environments tended to have much less lobing than plants from colder environments. And so these are just examples here of the different like lobing levels that we might see. And so this is a really strong correlation that Asia found both across all of the continental US, but also within specific regions. You can read more in her paper about this. But one thing we thought that was really cool about this finding was that work that Maya Wilson-Brown in the lab had done during the pandemic had showed that maximum temperature really matters a lot for where you find Capsella. So she'd done some ecological niche modeling of the North American range in Capsella. And she found that, maximum temperature contributes most. She modeled each month differently or independently and found that for most months in the summer temperature was really important for shaping the range. So we already knew that temperature was really important for these plants. And now we know it's important not just for whether they exist, but potentially for their phenotypes as well. Okay, so after finding that temperature mattered a lot for leaf shape, Asia took some of our New York City plants and grew them up in growth chambers at 3 different temperatures to try to understand the plastic response. Because what we'd seen in the herbarium data was that there's a difference related to the environment, but we don't know if it's because the plants are genetically different or if it's because they have plastic differences. And so Asia grew up all of these plants in growth chambers, different temperatures, and she found that there was a pretty significant relationship between temperature and lobing. So in particular, between 30 and 20, there wasn't a huge change, but the cold temperatures, so 16 degrees C, plants tended to be much less lobed than they were at 20 degrees C. Although there's some G by E from this. Some plants actually have less lobing and warm as temperatures go up. But in general, there's this overall significant pattern that plants are more lobed as it gets warmer, at least those cooler temperatures. So this was super duper puzzling to us because we did think that temperature was going to affect leaf shape, but we thought it would go the opposite direction. So we'd seen in our herbarium data was that plants from warmer environments were less lobed. But what we see is that when we grow them in the growth chamber, as it gets warmer, they become more lobed. And so we're currently doing more work to try to tease this apart, to try to figure out why is this happening? Is it that there's really strong counter gradient variation, where there's really strong differentiation between the populations? Something else. And so overall, for our in progress work on Capsella, what I've told you is that we see lots of genetic variation in small urban areas, and we are still trying to understand why. And we see that variation in nature differs compared to when we study plastic responses in lab environments. So my pitch is that it's important to look at both, not just one of those things. And then finally, we do still see evidence for genotype by environment interactions. So even plants from the same city respond differently to the same environment. That's, still don't know why, but stay tuned for more information about that. Okay, so I think based on time, I'm going to skip what else is going on in the lab, but I'm happy to talk to you all about it at another point because not everyone in my lab works on plasticity. So, okay, close your eyes, and that's not happening. All right. So overall, what I've told you about today is that hopefully I've convinced you that if we're trying to understand the maintenance of variation within populations and really big questions in evolutionary genetics, thinking about how the environment affects phenotypes is a really key component to that. And so, yeah, please, thank you for listening, and thanks to everyone in my lab who helped me with Dartmouth funding sources. Thank you all of you.
Time for questions.
Hey, really nice work. Have you heard that in the forest, comparing like northern forest with tropical forest, there is a tendency on the tropical forest to have like, how you say that? Like the leaves are not serrated, but in the temperate forest, leaves are serrated and more low. So that's a tendency that goes there. But I find it very strange that detail about that you find this latitudinal gradient, but doesn't correspond with the same thing in the growth chamber. Maybe it might be. I know that there is a hypothesis that it is a thing about cold physiology that makes leaves more serrated in the cold environments, but maybe photoperiod. Can you test that hypothesis data on the day of length, day length?
Interesting thing. We haven't tested that yet, but that would be interesting to think
yes,
about.
Because that's strange. I would expect that they were more serrated the colder they are.
Yeah.
But maybe.
I do think like talking to people who are leaf shape experts, there are these really broad latitudinal patterns that you can see. But then when, for like the evolutionary ecology world, people have looked at leaf shape variation within species at smaller scales, the patterns get a lot more complicated.
Yeah.
So part of it must be that like leaf shape affects so many different things, like in terms of not just temperature, but other things. So I don't know, but yeah, it's a good to think about. But like the scale, not tropical versus temperate, is going to be different than like within
Yeah.
region in the US. Thank you. Yeah, Ruben?
So on your first story, you were plotting only genes that show significant differences in expression.
Yeah.
And those were like only three
Only like
generations.
100.
So I'm just curious, do you think that over time, with more generations of selection, certain genes that didn't show difference in expression would become more significant, or what pattern what does your intuition tell you about how that pattern would change as you continue to increase selection?
Yeah, that's a really good question. I think, yes, we only had 3 generations of selection. And certainly those other experiments I told you about had more time because they were with, faster lived organisms. And yeah, certainly like maybe what was happening is we were getting response just based on sort of large effect genetic variation that's really common within the population. As we kept selecting, we might see different responses that are, over time. So it's a really good point, but I don't have a clear intuition other than I'm sure it matters a lot.
Has Gina continue evolving those populations?
I don't think she's continued evolving those populations, but I know someone who does evolve lots of population space, so maybe I'll talk to her. Yeah.
In the glyphosate experiment,
Yeah.
were the changes in gene expression in selected populations due to changes at the epigenetic level or the genetic level?
Oh, at the epigenetic level or the genetic level? I don't know for sure. Like we know that, like we were measuring them. They were measuring expression in the next generation after selection, but we don't know like if it's an epigenetic thing that was transmitted through generation. I don't know how to tell that right away. We haven't looked at methylation or anything like that.
Well, but lack of mutations, I don't know if you're doing only RNA-seq or
Oh,
if you're
yeah.
doing DNA-seq. If there's no mutations, then you can assume it has to be epigenetic, right?
Yeah, or I mean, there could be changes in allele frequency. But yeah, we tried calling, we only had RNA-seq data. We did try calling the SNPs using the RNA-seq data to sort of see if we could find evidence of like a selective sweep. But then we got worried that we had more, since we had more data for high expression genes, that would affect our power to detect a sweep. And so we didn't end up pursuing that. Thank you. Yeah.
So I'm not really in this field, but I wanted to just ask you to explain. You talked about the maladapt, the fact that if gene expression when you induce something is different to gene expression when you sort of evolve, you select for something, same
Yeah.
thing. If those two patterns are different, that then the plastic response is you would define it as maladaptive. I don't really understand why you would say that. You kind of just presented it as though that was obvious, but I don't really see that it's obvious.
I guess so. Maybe like thinking about not selection, but maybe replace that with like the shade avoidance pathway. Like let's say if you took a plant and you selected for doing well in a shade environment, over time you would expect to see the evolution of like, oh, if you're always in a shady environment, you should evolve that your response more constitutively. But if you, let's say if you took a plant and you put it in the shade and it actually, like got shorter in the shade and didn't do that, then that would be, the idea is that like it getting shorter is maladaptive because when you select over time, you see selection for taller height in like shady environments. So maybe it's easier to think about it for an actual trait. But like if you're seeing that like when I select for fitness and shade, they're getting taller over time. Then when I go back and see what they did in response to shade, they got shorter in the shady environment. That's kind of the opposite of what we know is improving fitness over time in that environment.
But I mean, improving fitness, in a particular, in response to a particular stimulus at a particular time, you may require a very different response than improving fitness, if that stress is applied consistently over multiple generations. You feel like there's just require very different responses potentially. And one could be adaptive in one situation, one could be very positive in the other situation. They both could be adaptive. At least I don't, unless I
Yeah,
don't
I mean,
understand
in
what
this
you mean
case,
by.
the stress is the same in terms of it being like the glyphosate spray was the same, and when we tested for plastic responses is when we selected for plants that did well in response to glyphosate stress. But you're right in that it could be more complicated in the sense that like and that's part of why Gina did those control environments, is that during selection for glyphosate resistance, you could also get selection for like higher fitness in whatever environment she was selecting in, which was a greenhouse in Michigan. And so like, that was why we did those control things, to at least show that like, it wasn't just the greenhouse, but it was the glyphosate that was responding. Hopefully that helped answer your question. I'll talk about it later. Yeah, Steven?
They talk, related to that question, so I'm curious of like the contrast between your findings of the prevalence of adaptive plasticity, so like the Gallup-Orr paper with prevalence maladaptive plasticity. And in the Gallup-Orr paper, they kind of conclude that evolutionary change is more likely in the presence of maladaptive plasticity, because
Oh, yeah.
adaptive plasticity would relax selection. So do you have a sense of what it means in your study system for evolutionary change?
That's a good question. So the idea is like, if there's some optimum and plasticity takes you in the environment, like let's say you're here and then the optimum in the new environment is here and plasticity gets you partway there, then selection is gonna be weaker to get you to the optimum than if plasticity takes you all the way over there. Yeah, that's a good point. And I hadn't put those two ideas together before, because Gina did find a response to selection in her trial. So I'm not sure if we know, is it like the plastic responses didn't quite get you all the way to what you would need to do, or maybe like, you know, that's one thing I would think about, or maybe just that the selection she did was strong enough to overcome that, because again, like she was doing quite strong selection because she wanted to get a response. I'm not sure, I'll have to think more about that. It could be that overall the studies are biased towards finding maladaptive plasticity, because that makes it easier to get that response you would need to do the study. Yeah.
Thank you for the talk.
Thank you.
There's this figure where you showed a bubble, where it you showed that accumulation in deleterious alleles might not be selected. Can it be that, there might be some kind of compensation happening for those deleterious alleles, and that's the reason it's not being selected, and that's why we're not seeing plasticity, because Arabidopsis is maybe a little simpler organism as maybe compared to maize, where maybe if there's a deleterious allele, it might not get compensated with other alleles. That's why we see more plasticity in maize as compared to Arabidopsis.
So if I'm understanding this, the idea is that like deleterious mutations in Arabidopsis and maize might be able to be compensated for by other stuff, and so then plasticity could kind of persist longer than in Arabidopsis, where there's sort of less like robustness because there's fewer genes in a smaller genome and stuff. Yeah, I hadn't thought about it that way before, but it's true. Like we're making a very simplifying assumption that all of these deleterious mutations are affecting fitness, and in different organisms, they might have bigger or smaller effects. Yeah, initially I had wanted to do this project in maize, and I don't actually remember why we did it in Arabidopsis, other than maybe the data was there was just more data that was available to us. So I could repeat it in maize. If someone wants to do all that work, but it would be even more genomic data.
Great, let's,
Thank you all.
Well,